Hey everybody, welcome to chapter 3. In this chapter we're going to talk about the cell and all the nitty-gritty details that keep the cells running and functioning. So what is a cell? A cell is the basic living structural and functional unit of the body. It is a single-walled unit that has its own functionings and instructions, and in some cases, in the non-human world, a cell can be an entire organism. Many bacteria are single-celled organisms. In our case, we have many cells that work together in order to make our organism. Okay, so let's start with the basics. We've got three main parts of a cell. We have the plasma membrane, we have the cytoplasm, and the organelles. The plasma membrane is the material that encases the cell and divides the inside of the cell and all of its parts from the outside world. The cytoplasm is the goo jelly that makes up the inside of the cell and carries all of the organelles. The organelles are the mini organ systems of the cell that keep the cell functioning and running. Okay, the plasma membrane. It is a flexible and sturdy barrier that surrounds and contains the cytoplasm. So its job is to maintain the inside versus the outside of the cell. It is comprised of what we call a fluid mosaic model. We call it that because it has lots of fluidity to allow the proteins that are embedded in it in order to move around and shuffle. It allows things in and out of the cell while being strong and keeping certain things out and certain things in. Okay? It is comprised of phospholipid molecules. Okay? It has a polar phosphate head and a nonpolar lipid tail. That head area is a hydrophobic head comprised of the phosphate and a glycerol, just like our saturated and unsaturated fats. And the hydrophobic water-hating tail is comprised of both a saturated and unsaturated fatty acid tail. Okay, so these phospholipids, sorry, I lost track here. Comprise together to make a phospholipid bilayer. So bi means two, so it's a double phospholipid layer that work together and forms the plasma membrane. Okay, so this phospholipid bilayer, naturally due to the chemical reactions, has these hydrophobic tails naturally bonding together, and the hydrophilic, the water-loving heads sticking out either on the intracellular, the inside of the cell, or the extracellular space, which is the space outside surrounding the cell. Okay. Here's a great model of this fluid mosaic that we were talking about. So we've got all of our phospholipid heads and tails bobbling around, and we've got a lot of these protein structures floating around the plasma membrane doing different jobs. Okay, we've got peripheral membrane proteins that hang out. We've got integral protein members. We've got lots of cholesterol for structure and support. We've got lots of protein channels that allow things to flow in and out. And we've also got lots of signaling molecules attached to the outside. Here's just another great way to look at the plasma membrane. So the plasma membrane of the cell is very complex, and it is comprised of lots of different proteins that make up and help the cell function. Okay, so the function of the plasma membrane is to regulate passages of substances in and out of the cell and to divide the inside from the outside. Okay, we talked about how it's made up of this lipid bilayer of the two back-to-back phospholipid heads. Here's that image again. Okay, components of the phospholipid bilayer. So most of the plasma membrane, is made up of these phospholipids that we talked about. It is going to allow it to have a polar and non-polar parts. The plasma membrane's also made up of about 5% glycolipids. These are really important for adhesions. That's going to allow cells to connect to other cells, cells to communicate with other cells, and it will help regulate growth and development. So when one cell dies, it's these glycolipids that are going to allow and signal the cell to then multiply and reproduce to fill up that gap. Okay. We also have cholesterol. This is parts of the bilayer, the plasma membrane, that's going to aid in strengthening and supporting it. We want it strong, but we don't want it too strong. But if it's not strong enough, it'll collapse on itself. We have two different types of membrane proteins in our plasma membrane. We have integral proteins, which extend across the entire phospholipid bilayer, and peripheral, that only extend part ways. Okay, so we have got, again, the integrals that are going to extend the whole way, and this is actually a peripheral, okay, that's going to just be on the end. Okay, what are some of the functions of all of these proteins? So we talked about we've got all these different types of proteins in and around and on the plasma membrane. So why are they there? One reason is transportation. We need to be able to get things in and out of our cells. We have different membrane proteins that can move goodies in and out. Some require energy in the form of ATP. Some do not. And we'll go into that a little bit more. Okay. Some of the membrane proteins are there for signal transduction. They are going to allow the cell to communicate with the exterior environment and let the environment tell the cell different things. So a good example is hormones are going to be able to signal receptors on these cells to make the cell respond or create different proteins or different products depending on the signal. Okay. We also have attachments to the cytoskeleton and extracellular matrix. The extracellular matrix is all of the stuff on the outside of the cell. We need to make sure that our cells can attach to the exterior environment and anchor when needed. If we didn't have anchoring proteins in our skin, they wouldn't be attached to anything, and you could literally peel off our skin, and that would be gross. Okay. We have special proteins that have enzymatic activities. Sometimes we need enzymes in order to catalyze chemical reactions, and we want that done at the cellular level or on the plasma membrane, maybe because some of the materials are only found on the outside, or maybe that's just a great place to anchor these enzymes. But we've got quite a few different types of enzymes that help catalyze or speed up chemical reactions found in the plasma membrane. We have intracellular joining, so we need some way for two cells to connect to each other. We want some of these cell-to-cell interactions and connections so that the cells can stay bonded and communicate when necessary. And we have cell-to-cell recognition, so we need to be able to recognize self. We need to recognize what cells belong to us and what things are a foreign body. These glycoproteins also float around in the plasma membrane. So lots and lots of different things going on in the plasma membrane. All very complex and all very important. Okay, so let's talk about permeability of the membrane. Permeability, how much passes through, in, and around the plasma membrane. The plasma membrane is going to permit passage of some things and not the other, making it selective permeability. Okay, so the cell, via the membrane, can determine what things it wants to bring in, what things it wants to let out, and what things it just wants to communicate through these signaling molecules. Okay, one of the reasons that it wants to do this is there's often the need to maintain a concentration gradient. So we want often different amounts of materials on the inside of the environment compared to the outside of the environment. If we had the same amount of molecules on the inside and as the outside, there'd be no need for the plasma membrane. Why not just have all the organelles floating out and around? But with the plasma membrane, we can allow molecules and charges to be different on the inside of the cell as in the outside of the cell. A really great way to think about this is saltwater fish. Okay, what would happen if the saltwater concentration was the same on the inside of the fish cells as the outside? Saltwater makes you want to retain water, and so we need to make sure that we can keep the salt in the salt water out and the proper amount of water on the inside in. Or in humans, a common problem we have is dehydrating, and so we want to make sure that we have more water inside than the outside. Okay? We also are going to use different ions to maintain different charges on the inside and the outside, and we're going to talk about that a little bit more. Okay, so there's multiple different ways that we can get materials to cross the plasma membrane. Some of them are going to require some of those membrane proteins, but not everything. One way that is a super easy way to get things across the plasma membrane is through just diffusion. Okay, so diffusion is the passive, the non-energy requiring movement of small molecules through the cell membrane down their concentration gradient. Okay, so if you have a whole bunch of teeny tiny little molecules, such as oxygen or carbon dioxide on the outside, they're going to just naturally percolate or trickle through the plasma membrane into the cytoplasm because it wants to maintain equilibrium on both sides of the plasma membrane whenever possible. So if it's small enough, it's just naturally going to filter and bop its way through these phospholipid bilayers in order to maintain an equilibrium here. Not everything can do that, but small items are able to do that. And this is great because it does not require any energy in order to allow this to happen. This is just simple chemistry. Okay. Another way that materials can get through the bilayer is through facilitated diffusion. Okay, so facilitated diffusion, it requires a membrane protein in order to cross the plasma membrane. It can't do it because it's too big to go through on its own, but it can utilize a channel protein in order to cross into the cell. Okay, these carrier proteins are more selective and often only carry one particular type of molecule per channel protein. Okay, so you have these proteins that will only allow its specific molecule to pass through. It can still use concentration gradients for some of these larger molecules, but it's going to only allow one type through and not everything through. Okay, again, we want to maintain some different concentration levels, and so we don't want to let everything into our cells. Okay, I talked about the charges. Sometimes we want to maintain different charges on the inside and the outside, and eventually we'll talk about how we utilize these charges for chemical reactions and signalings. So ions are a great way to maintain different charges. So here's an example of allowing certain ions to transfer through to keep the outside of the cell positive and the inside of the cell negative. And this can be done again through these facilitated diffusion during the channel proteins. Okay, so we've got essentially two different types of movement of materials across the plasma membrane. We've